Hello everyone, welcome to Chemazon-Complete Chemistry. So in today's video, we are going to learn some of the very important terminologies based on amino acids, peptide bonds, what are the different types of peptides, what is C-terminal, N-terminal, and what is Zwitter ion. Okay, so let us start with what is exactly a Zwitter ion. So till now what we have drawn, the structure of amino acid, it has one amino acid, one amine group, then there is a carboxylic acid group, okay, then there is hydrogen, and then there is some side chain, let's say R. Okay, so this is the structure that we have drawn till now. Is this correct? The answer is no, this structure doesn't exist. Okay, naturally, if you see, the amino acid does not exist in this form. Why? The reason is very simple. Carboxylic acid, it has a tendency to donate what? It has a tendency to donate H plus ion. Okay, it will donate H plus ion and it will become carboxylate form. Okay, carboxylate form. Okay, and then it has donated H plus ion. Where will this H plus ion go? To the amine group. Okay, this will pick up this H plus ion and it will be protonated. Okay, NH3, this will become NH3 plus. So this is what is the form that exists. This form of the Zwitter ion, this form of the amino acid is called as a Zwitter ion. Okay, Zwitter ion. What is the meaning of this term Zwitter? It means mixture of charges. Okay, it has both positive and negative charges. Okay, so overall charge, if you see, overall charge of a Zwitter ion is always going to be 0. Okay, overall charge is always 0. Okay, in the next video when we study electrophoresis and try to understand isoelectric point, that time you will understand what is the exact meaning of this Zwitter ion having 0 charge. Okay, so this is a structure of what? This is a structure of the Zwitter ion. Okay, in the naturally it occurs, all the amino acids occur in the Zwitter ionic form. Okay, so what is the next topic is what exactly is peptide linkage or peptide bond. So I had told you, if you remember, proteins are what? Proteins are biopolymers. Okay, proteins are what? They are type of a biopolymer. Biopolymer means naturally occurring polymer. Bio means life, polymer. So it is a polymer that is present in living beings. Okay, we can say naturally occurring polymer. So we know if it is a polymer, it will definitely have some small repeating units or monomers. Okay, so what is a monomer? We know that is what? That is amino acid. Amino acid is the monomer of proteins. So what exactly happens is, suppose I take one amino acid A1. Okay, I will take another amino acid A2. I'll take another amino acid A3. They keep on linking to one another, and in this manner we will get a very long chain of amino acid that is called as a peptide chain. That is called as a peptide chain. Now the question is, what is the exact meaning of this peptide chain? Okay, suppose I take this amino acid. Okay, let me erase this. Okay, I take this amino acid. What is the name of this amino acid? Side chain is hydrogen. So this is glycine. Okay, and I want to connect it to some another amino acid. Let's say this. What is the name of this amino acid? If this was CH3, I told you what it is called. It is called alanine. Here it is phenyl group, is one hydrogen is substituted by phenyl group. So this is what? Phenylalanine. What is a 3-letter word? PHE. Okay, so what exactly happens is, two amino acid molecule will combine. There will be loss of a water molecule like this. Okay, water molecule will be lost, and draw the entire structure. Okay, I'm teaching you to draw the peptide chain. Here there are two amino acids, so you draw whatever is remaining. Okay, NH2, CH, H, C double bond O, then here NH is remaining, CH, CH2, benzene ring, C double bond O, OH. So this is what this is called as a, this is made from two amino acids. Okay, so this is called as a di. Di means two, peptide. Okay, dipeptide means a compound or a peptide chain having 2 amino acids. Okay, suppose I write tripeptide. What is the meaning of a tripeptide? What is the meaning of a tripeptide? It consists of 3 amino acids. Okay, if I tell you a tetrapeptide means it has how many amino acids? 4. Suppose I write pentapeptide. What is the meaning of pentapeptide? It has 5 amino acids. Okay, and so on. If it has multiple amino acids, okay, linked together, then at the end we call that as polypeptide or proteins. Okay, we'll understand exactly how we make proteins from this peptide chains. So I hope you have understood till now how do you name if 2 amino acids dipeptide, 3 amino acids tripeptide, and so on. Okay, now the question is, what exactly is a peptide linkage? So this amide bond CONH, this is what this amide bond is nothing but called as a peptide bond. Okay, this amide bond, it is called as there are two names. It is called peptide bond, or it is also sometimes called as a peptide linkage. Okay, linkage. Okay, here in amino acids we have what? We have amide bond or peptide linkages. In sugars we will see it has ether linkage, that is nothing but called as glycosidic bonds. Okay, that is called as glycosidic linkage or glycosidic bond. Okay, that we'll see later on. Here in proteins, what we call it as peptide linkage or peptide bond. Okay, now the question is dipeptide. Okay, this is very important. The name is dipeptide. Okay, does that mean there are 2 peptide bonds? No. Okay, dipeptide, does that mean there are 2 peptide bonds? No, that will be incorrect. This is the structure of dipeptide. You can see there is only one peptide bond or one peptide linkage. Okay, so how do you find out the number of peptide linkages? The number of peptide linkages is written as n-1, where n is what? N is the number of amino acids. Okay, dipeptide means there are 2 amino acids. So dipeptide, there are 2 amino acids, so 2 minus 1, there will be 1 peptide bond. If I say tripeptide, there will be 3 amino acids, 3 minus 1, there will be 2 peptide linkages. Suppose I give you a question, okay, a hypothetical question. Let's say there are 100 amino acids, and the question is how many peptide bonds are present. So the number of peptide bonds will be 100 minus 1, that is 99. Okay, so in this way you can find out the number of peptide linkages. Okay, so this is an example of a dipeptide. Okay, glycine and alanine, they combine, there will be loss of water molecule, and we will get how do you name a dipeptide? Okay, first amino acid you write first glycine, and then second amino acid you write alanine. So just like methane, okay, for methane we write what? Methyl. Similarly INE, okay, last 3 letters you will replace and you will write what? YL. You will add YL. Methane becomes methyl. Similarly glycine becomes glycyl alanine. Okay, so this is one dipeptide. Now suppose I switch this, alanine I write first and glycine I write second. Will that dipeptide will be same or different? The answer is it will not be same, it will be different. Alanine is first, then glycine. This is a different dipeptide. Now you write alanine first, then you write glycine is second. So you write glycine. First one you remove INE. What you will write? You will write YL. So alanyl glycine. Okay, alanyl glycine. And how do you write the 3-letter word? Alanine is ALA. This dash means it is what? It is a peptide bond. Okay, then GLY. Okay, so in this way you write the dipeptide or polypeptide in the 3-letter notation. Okay, so how many peptide bonds are present? Only one is present. Okay, 2 amino acids, so n-1, so 2 minus 1, there is only one peptide linkage. Okay, now this is what I've taken. Now I've taken the dipeptide which I had formed, alanine, alanyl glycine, and I'm adding one more amino acid. So what I will get? I will get a tripeptide, alanine, glycine and lysine. So in this way, to this tripeptide, if I add one more amino acid, I will get tetrapeptide. To that, if I add one more, I will get pentapeptide and so on. Okay, in this way, multiple amino acids will keep on joining, and in further video, we'll see there are different structures of proteins that are formed. Primary, secondary, tertiary, they will recoil, coil, recoil, coil, recoil, and at the end, we will get protein. So that we'll see at the end. Okay, so this was tripeptide. Now this is a homework for you. What you have to do is you pause the video, they count the number of amino acids. Okay, the question is what you have to do is, the question is, I want you to draw the structure of the peptide chain. Okay, draw the structure of the peptide chain. I want you to find out the number of amino acids that are present. Okay, number of peptide linkages. Okay, and write it in the 3-letter notation. Write the name of that in the 3-letter notation. Write the name of that polypeptide or peptide chain in 3-letter notation. Okay, so what exactly happens? You can pause. I will tell you the answer. Okay, water will be removed. There are total how many amino acids? There are total of 5 amino acids: alanine, serine, glycine, cysteine, valine. Okay, so 4 water molecules will be lost, and this is the final structure that we get. Okay, I will erase this. So how many peptide bonds are present here? How many peptide linkages are present here? 1, 2, 3 and 4. So if you use the formula, that also it is fine. How do you find the number of peptide bonds? N minus 1. Amino acids is 5, N is number of amino acids. So 4 peptide bonds, that is correct. If I want to write down the 3-letter notation name of this, what will this called as? This will be called as a pentapeptide. Pentapeptide. Okay, this tetrapenta, what does it indicate? It indicates the number of amino acids. Okay, number of amino acids and not the peptide bonds. Number of pentapeptide, 5 amino acids. Okay, if I want to write down the name, first it is alanine. Alanine is ALA. Then we have serine. Serine is SER. Glycine is GLY. Cysteine is CYS. Valine is VAL. So this is the 3-letter name for this pentapeptide. So this is the end of this. Okay, so one more topic. We didn't discuss what is N-terminus and C-terminus. So if you see all the peptide chain, I have started with amine group. Okay, then in between there is carbon, hydrogen, R group. Okay, then we have NH. So if you see, the peptide linkage always starts from carbonyl, that is CO, and ends with NH. Okay, CONH, carbonyl group, and then NH. So if you see the first amino acid, which is the group that is left out, it is always going to be the amine group which is present on the left. Okay, and you will see when you draw, always carboxylic acid is a group that will be present on the right. So the end that has amine group, that is called as a N-terminus. Free amine group is called as a N-terminus, and on the right, the chain, part of the chain which has free carboxylic acid group, that is called as the N-terminus. Okay, so N-terminus has what? N-terminus has a free amine group. C-terminus has a free carboxylic acid group. Okay, so in this way, here you can see on the left, this end is called as the N-terminus, and the right one where it comprises of the carboxylate ion, that is called as the C-terminus. These are the peptide bonds we already know. Okay, so now this is the end of this video. In the next video, we will learn a separation technique. Suppose there are a mixture of amino acids, and if I want to separate them, which technique we can use? That is called as electrophoresis. There we will understand what is isoelectric point and how do you calculate isoelectric point for different amino acids. Thank you.